Humans are sexually reproducing viviparous, continuous breeder animals. Yes, I'm talking about the chapter human reproduction and the topic gametogenesis, the most important one. And if you observe the previous year's questions of NEET, mostly repeated questions we're gonna get from this particular topic from human reproduction, that is gametogenesis. My dear lovely students, this is Biswajit, your zoology mentor, and today in this session we're gonna complete or cover this particular topic with proper conceptual aspect as well as with practicing few of the important questions from this particular topic. So let's start. The subtopics we're going to cover under these, they are the definition of gametogenesis, spermatogenesis, oogenesis, regulations of gametogenesis and oogenesis, and obviously the distinct difference between the course of spermatogenesis and gametogenesis. Okay, so if you're asked that define gametogenesis, now if you consider the most important events of human reproduction, they're like this, starting from formation of gamete, that is called gametogenesis. Then after that, transfer of sperms or male gametes into the reproductive tract of female by the process of insemination, and third step, that is fusion of male and female gametes to produce diploid zygote, the first cell of a life. And the fourth step of the events, if you consider the events of human reproduction, then that is the formation of embryo and implantation of that into the uterine wall, right? Okay, and upon this implantation, gestation period or embryonic life starts within the mother's womb, and after 9 months there will be the phenomena of the delivery or the childbirth or the termination of reproductive event with the process of parturition. Now in this particular session, obviously we will be focusing particularly on these important phases of gametogenesis. So this is the process of formation of gametes. Now what do you mean by gametes? Gametes are haploid cells. What do you mean by haploid cells? They have only n number of chromosomes. Now what do you mean by that? So if you consider a somatic cell of our body, a normal cell of our body, you will be finding that has 23 pairs or 46 chromosomes present into that. Now whenever our body is forming these haploid cells or gametes, you will be finding only 23 chromosomes into that. That means it's half of the normal somatic cells. That's why those somatic cells are called as diploid or 2n, and these gametes are called as haploid or n, right? Now in case of males and females, you will be finding two different kind of gametes. For male, you are gonna get sperms or spermatozoa, and in case of female, you are gonna get ovum, right, as female gamete. So these are considered as gametes produced by male and female. Okay, now why it's important? Why do we need to study about this process of the whole mechanism of gametogenesis? You know that these gametes are produced inside the gonads or the primary sex organs. In case of male, these sperms will be producing inside the testes or testis, and in case of female, the ovum will be producing inside the ovaries, right? Now, from which particular cells they are developed? From gamete mother cells. We call them germinal cells. Now, this, if you consider these germinal cells, they're just like somatic cells of your body. They are diploid in nature, but upon undergoing some sets of division, they can form a special kind of cells with half of the numbers of chromosomes. Now, why these gametes are posing only 23 chromosome? Why they don't have 46 chromosomes? Suppose you have to undergo fertilization between two gametes, right? You have to undergo fertilization between sperm and ovum. Now, if the sperm already is having 46 chromosome and the ovum is also have the this 46 chromosomes, upon fusion, the zygote and the new embryo and the new individual is formed, that each cell of that particular person or that newborn child will be posing 92 chromosome, 46 plus 46. Now that will give rise a new kind of species that won't be a human being, right? But obviously we know the importance of reproduction. The child of human being should be a human being only. For that we have to maintain the chromosome number. And how to maintain that? If you making the chromosome number half inside sperm and ovum, that's 23 chromosomes inside sperm and 23 chromosome inside ovum, the resultant zygote gonna get 23 plus 23, 46 chromosome, just like the normal cells of our body. And that is the importance of gametogenesis and why these gametes are posing only half of the chromosome in compared to our body cells. Okay. Now let's move forward for the process of spermatogenesis. Okay. So spermatogenesis is taking place inside male bodies. The primary reproductive organ called testis. So here you can observe a sectional view of testis. These are the testicular covering, we call it tunic, and inside each testis is divided into chambers called testicular lobules, and inside a single testis you are gonna get near about 250 chambers, right? And inside these chambers, this highly coiled tubular part can be seen. We call them as seminiferous tubules, and inside these seminiferous tubules the mechanism of sperm production going on, and that mechanism, the whole lot of mechanism happening inside these highly coiled tubes, we are calling that as spermatogenesis, the process of male gamete production or sperm production, right? Okay. Now for that we need to maintain a particular temperature inside our testis. Now if this is a human body, you know that one pair of testes present inside skinny bag called scrotal sac or scrotum, and inside that scrotum one pair of testes are situated, right? Now why this primary sex organs of males are situated outside the body? We call them extra abdominal organ, because you know that the normal body temperature is 37 degrees Celsius. but this is not optimum for the production of sperm or for the process of spermatogenesis. For that, we need -2 to 2.5 degree Celsius in compared to our body temperature. So basically for sperm production, we need 34 to 35 degrees Celsius, which is not possible inside our abdomen or inside our body. That's why during the time of our embryonic development, the testes, uh, like one pair of testes, they will be descending outside the abdomen and situating inside the extra abdominal sacs called scrotal sacs or scrotum in order to maintain -2 to 2.5 degrees Celsius temperature. Okay, and what are these particular structures? You need to know their name. So this is a diagram of male reproductive system. We are not gonna study in detail about this. So this is one pair of testes, left testis and right testis, and they are inside scrotal sacs, and outside the testis highly coiled tubes are called as epididymis, and after epididymis we are gonna get these sperm ducts or vas deferens, right? Okay, and inside the testis, the highly coiled tubes present inside the testis, uh, one, two, or three inside each testicular chamber or each testicular lobule. These tubes are called as seminiferous tubule. And these seminiferous tubules, why they are so much important for us? Because they are the site of spermatogenesis. Okay, now let's take a look for the process of spermatogenesis. Now before starting this, we need to talk a little bit about the seminiferous tubules and all. So if this is cross section of a single seminiferous tubule, we're gonna get special kind of cells into this, and these particular cells are responsible for sperm production. We call them germinal cells, right? These particular cells are germinal cells present inside seminiferous tubules. Now they need special stimulus to start the process of proliferation and maturation, and from where they're gonna get this stimulus? Outside the seminiferous tubule, this space is called as interstitial space, and into this interstitial space we'll be finding some special cells are present, and these particular cells are called as Leydig cells. Leydig cells, or you can also call them interstitial cells, is because these cells are situated inside the interstitial space. Now what do you mean by interstitial space? The space outside the seminiferous tubule. What is this? This is our seminiferous tubule. Okay, inside this seminiferous tubule, sperm production will take place, and the signal will be coming from outside the seminiferous tubule from those Leydig cells. Okay, now who will be stimulating the Leydig cells? There is a hormone which is secreted by our pituitary, that is called luteinizing hormone, LH, a hormone of our anterior pituitary. So this hormone gonna stimulate these Leydig cells, and these Leydig cells gonna secrete some male sex hormones. We call them androgens. Now, you know about testosterone, right? Now, testosterone is one of the prime androgens produced by the Leydig cells. Now, these androgens will be called inside seminiferous tubules. And under influence of these androgens, and obviously few more things will be stimulating them. Here inside seminiferous tubule, you are going to get this kind of tall, slender cells as well. We call them nurse cell or Sertoli cells. So under influence of some special factors secreted by these Sertoli cells and androgens or testosterone, gradually the cells will start proliferation and gradually they will be forming sperms or spermatozoa inside the seminiferous tubules. Proliferation means gradually the cells are dividing to form mature spermatozoa or sperms. Right? So this was the overall phenomena. Let's check inside the seminiferous tubule how these germinal cells are getting proliferated or matured into spermatozoa. Spermatozoa is another name of sperm. Okay. Now these germinal cells, they will be modified into these germinal cells. They are modified into special sperm forming cells called as spermatogonia or a singular we are calling them spermatogonium. Now this spermatogonia, they are also diploid in nature. They have 46 chromosomes inside them. Now upon our puberty means adolescence, these spermatogonia cells will start proliferation inside the seminiferous tubules of testis and they will form some similar kind of cells by the process of mitosis, right? They will form more spermatogonium. Now in plural we are calling them as spermatogonial cells. Obviously, do not forget that these are diploid in natures. Now from this particular spermatogonium, there will be a special kind of modification. We call it as differentiation. Now some special spermatogonia, they will undergo differentiation and they will be forming an enlarged version of spermatogonia and now we'll be calling it as primary spermatocyte. Now as because this is only mitosis and differentiation, there won't be any change in chromosomal number. So these primary spermatocytes, they're also diploid in nature only. Now from here the real process is beginning. Now what will happen? We need to uh, decrease the chromosomal number into half, right? For that, we need to perform a special kind of cell division and we call that reductional kind of division as meiosis, right? So, from primary spermatocyte, there will happen a special kind of a cell division because we call it first meiotic division. Okay. Now, as a result of first meiotic division, from one single primary spermatocyte, we are going to get two secondary spermatocyte. Now, as because this first meiotic division or meiosis 1, we know that meiosis will be taking place into two steps, first meiotic division and second meiotic division. The first meiotic division is a reductional one. It's a reductional type. That means chromosome number will decrease now into half. Now this secondary spermatocytes are possessing only 23 numbers of chromosome inside them. And that's why we are calling it as a reductional division. So first haploid cell produced during the time of spermatogenesis, they are secondary spermatocyte, they are not primary spermatocytes. Okay. Now what after secondary spermatocyte? Obviously meiosis has begun, but we have to complete the meiotic division. Now there will be beginning of second meiotic division. And you know that second meiotic division is a kind of equational division. That means there won't be any change in chromosomal number. So from secondary spermatocyte, from one secondary spermatocyte, two new cells are formed and we'll be calling them as spermatids. Spermatids. Now as because they are resultant cells of a second meiotic division, there will be no change into their chromosome number. So spermatid, they're also possessing 23 chromosome inside them. So from one primary spermatocyte, we will be getting two secondary spermatocyte, and each of the two secondary spermatocyte will undergo second meiotic division to form four spermatid. But if you are asked that how many spermatids are formed from one secondary spermatocyte, from one secondary spermatocyte, two spermatids are formed, but from one primary spermatocyte, four spermatids are formed here. Okay. Now what after spermatid? Now here the cell division process is stopped. Now there will be structural modification of this spherical spermatid cells. Now we need to do what? Structural modification. The spermatids are spherical in nature. Gradually they will start changing their shape and will be modified into tadpole-like cells called as spermatozoas. That is called structural modification, right? Or structural maturation. So here we need help of some chemical agents which will be secreted by the Sertoli cells for this last step, structural maturation of spermatid. Here we need stimulus of a special kind of chemical called as SMF, sperm maturation factor, and this sperm maturation factor is secreted by what? Sertoli cells, these elongated cells present inside the seminiferous tubules, and under the influence of sperm maturation factor, the spermatids will undergo differentiation, means structural modification, and will be getting a new cell which has similar chromosomes like spermatid but with the new design, and we call them as spermatozoa. they are regarded as male, mature male germ cell or sperms. So this was about the whole process of spermatogenesis, right? From spermatogonium to spermatozoa, the whole process is called as spermatogenesis. But the last phase from spermatid to spermatozoa, the only differentiation part from spermatid to spermatozoa, this stage is called as spermiogenesis. So what is the difference between spermatogenesis and spermiogenesis? Spermatogenesis, the whole process, the sequential steps of formation of spermatozoa from spermatogonium, the whole process. But the last step of spermatogenesis, where spermatids are getting differentiated to form a structurally mature sperm called spermatozoa, which are also haploid and with 23 chromosomes. This last step is called as spermiogenesis. Got it? So we are done with the process of spermatogenesis. Now we'll be moving forward. Okay. Now few more terms you are gonna get, and they are quite confusing. First is spermatogenesis. Second is spermiogenesis. And the third terminology is spermiation. Now, multiple times this kind of terminologies are asked to elaborate. Now, spermatogenesis, the whole process of sperm formation is called spermatogenesis. Basically, from spermatogonia through sequential steps and ultimately formation of spermatozoa, that is called spermatogenesis. Spermiogenesis, the last step of spermatogenesis, from spermatid to spermatozoa. This is not a cell division, this is only differentiation, means structural modification in cells. Simply this spermatid will be modified into sperms. And the third terminology, spermiation, it's a different thing. Now, what do you mean by spermiation? Now we know that inside the Sertoli cells, we have some nourishing cells, this elongated kind of cells called as inside the seminiferous tubule, we have these Sertoli cells. Elongated cells are called Sertoli cells. They are present inside into the seminiferous tubules. Okay. Now, after the formation, you will be finding these spermatozoa. They are embedded into the crevices of this Sertoli cell. Once they are structurally matured, they will be released from here. They will be released from here. They will be released from here and they will gradually proceed outwards. So this particular process of release of mature spermatozoa from the crevices of Sertoli cell and then into the lumen of seminiferous tubule, and gradually that's coming out through the secondary reproductive tract of human reproductive system, that process is called as spermiation, right? So spermiation is not included inside spermatogenesis. Once spermatogenesis is done, the process of release of the structurally matured sperm, the process of release of structurally matured sperm from Sertoli cell into the lumen of seminiferous tubule, and then through the progression through the secondary tubular part of our male reproductive system, that whole process is called as spermiation. Okay. Now here you can see the diagram of spermatozoa or sperm. So it's very simple. The structure of spermatozoa, if you consider this whole structure, it has head, neck, middle piece, and a flagellum or tail. So these are the four parts of a mature spermatozoa: head, neck, middle piece, tail or flagellum. Now what is the importance of head? It's quite compact in nature. The shape of the size of head is near about 5 micrometer. It has a modified Golgi complex called acrosome. What is acrosome? It's basically modified Golgi complex. Modified Golgi complex. Now we know that Golgi bodies and Golgi complexes, they are responsible for modification of protein, and these proteins are modified into enzymes. So basically these acrosomes are nothing but reservoirs of hydrolytic enzyme. Now why you need this acrosome, this cap kind of structure at the edge of spermatozoa? With the help of this hydrolytic enzyme, we can call them as sperm lysins. Lysin means the enzymes which are responsible for breaking down the substances, right? So this sperm lysins present into the acrosome, they are used during the time of fertilization while the sperm is penetrating inside the secondary oocyte, right? To penetrate, you need to dissolve or break down some chemical bonds of the membranes outside the ovum, and here at that time the sperm gonna utilize this kind of sperm lysins present inside the acrosome. Okay. Now a compact nucleus present into this head region which has 23 chromosomes. Let's come to the neck part. The neck of sperm has two very important cell organelles. Centrioles are there. So one proximal centriole and one distal centriole. This one is proximal and this one is distal. This distal centriole, this is responsible for the formation of the flagellum or the tail of sperm, and this proximal centriole that is responsible for the zygotic cleavage. This is the particular centriole which will be donated to the ovum after the process of fertilization, right? Okay. Now let's come to the middle piece part. Middle piece part is rich in ATP producing mitochondria. Now why you are going to observe mitochondrial spiral, not thousands, hundreds of mitochondrions are present here, and these are energy source for swimming, right? When the sperm will be swimming or swarming towards the ovum, aiming for fertilization, they needs energy. Now, this mitochondria will be utilizing the energy source, and the main energy source for sperm is gonna be fructose present into the semen, right? Okay, and with the help of tail, they will perform swimming or swarming. So the energy source will be the mitochondria present into this middle piece of sperm, and with the propulsion power capability of this tail or flagellum, they will be swimming towards the ovum, aiming for fertilization. Okay, now we are done with the process of spermatogenesis. Now we'll talk about oogenesis. Now, in order to understand the whole process of oogenesis or ovum production, we need to get a brief idea of female reproductive system, right? So this particular portion where implantation will take place, that is called as uterus, and this particular cavity is called uterine cavity. This arm kind of structure, that is called fallopian tube or uterine tube or oviduct, right? This is the particular place where fertilization gonna take place. This outer part is called as vagina. After insemination, a male through the help of copulatory organ penis gonna deposit semen inside this particular portion, and the neck-like structure which is connecting the vagina to uterine cavity, we call it as cervix, right? And obviously the both side of uterine wall, you are gonna get two ovaries, and these are the primary reproductive organs of female. Now, what do you mean by primary sex organ or primary reproductive organ? They are responsible for gamete formation. Gametogenesis is happening inside them, and also they are responsible for the production of the sex hormone. Like for if we talk about the testes, they are primary sex organ of male, and they are responsible for sperm production and production of male sex hormone androgens or testosterone. For female, these ovaries are considered as primary reproductive organs, as because they are responsible for oogenesis, ovum production, also for the production of female sex hormones called estrogens and progesterone. So that was brief about the female reproductive system. Let's move forward uh, to study in detail the process of oogenesis. Okay. Now, if you study in a proper manner, if you get inside the uh, ovary, one single ovary, it's divided into different zones, right? First, there will be ovarian surface epithelium. There will be one layer of fibrous tissue, that is tunica albuginea, below the ovarian surface epithelium, and inside the ovary you are going to get this part is called as stroma, ovarian stroma. Now this ovarian stroma is divided into two sections. The inner zone is called as ovarian medulla, and the outer zone of ovarian stroma is called as ovarian cortex. Outer zone of ovarian stroma is called as ovarian cortex. And the inner zone of ovarian medulla ovary is called as ovarian medulla. Now, what is the importance of ovarian medulla? This is the zone where blood vessels, nerves are entering through it. Which zone? Through medulla. And cortex is the highly entropic zone where you can observe this process of oogenesis happening along with the development of the ovum containing cages. We call them as ovarian follicles, right? So this process of oogenesis or maturation of ovum happening inside some ball kind of structure, these glandular structures, and they are called as ovarian follicles. And here I have shown the maturation stages inside the ovary or the cortex region of ovary, and you can observe the follicle will start from the immature one called as primordial follicle, and after primordial follicle there will be formation of primary follicle, and this formation of primary follicle will be taking place during the time of embryonic life or embryogenesis for a female, and that is the difference in the phenomena of spermatogenesis and oogenesis. In case of male, spermatogenesis starts at the time of puberty or adolescence at the age of 11 years or 12 years or 13 years. But in case of female, the process will be initiated during the time of embryonic life only. You need to mark that difference, right? So inside the ovary, when a girl child is not parturited yet, she is lying inside the mother's womb. Her ovaries will be activating the process of oogenesis. There will be formation of primary follicle, and that is written here as 1-degree follicle. 1-degree follicle means primary follicle, and inside that primary follicle you are going to get one immature stage of ovum, and that is called as primary oocyte. That is called as primary oocyte, right? Now how these primary oocytes are formed? They are produced from oogonia, right? The first cell from where the spermatogenesis started, we call that cell as spermatogonia. Now the first cell from where this oogenesis will start, we call that as oogonia, and the developed one, the developed stage of oogonia is called as primary oocyte, which you are going to get inside this primary follicle, and everything is happening during the time of embryonic life of that girl child, right? Okay. Now, how many primary oocytes are present inside the ovaries? Millions. Billions of primary oocytes are present inside the ovaries. But the actual reproductive life will start from the age of adolescence or puberty at the age of 11 years or 12 years or 13 years. At that time, you'll be finding only 60 to 80 thousands of primary oocytes are left inside the ovary, right? So during this gap of 12 to 13 years, starting from the embryogenesis, inside the primary oocyte, you'll be finding one arrested form of this, right? Inside the primary follicle, arrested primary oocyte you are gonna get till the time of puberty. Now why it's so? See, if you study the phenomena, it's like this. Starting will be from oogonium. After oogonium, you are gonna get primary oocyte. After primary oocyte, you are gonna get secondary oocyte. After secondary oocyte, you are gonna get the matured ovum. You can also call it as oocyte. So these are the different stages of ovum. Now I am saying oogonium to primary oocyte conversion will be there into their embryonic life itself for girl childs. So once the biological environment inside the embryo of the girl can detect that it's only few months of conception and the little child is already initiated the process of oogenesis, they will like they will be scolding that primary oocyte into chemical manner with the help of some chemical agents, and they will be arresting further division of primary oocyte. And when after birth, gradually the girl child will grow up and reach to the adolescence or puberty at the age of 12 or 13, then again due to the influence of some special pituitary hormones, the development or the division of this primary oocyte will start, and that will be modified or converted into secondary oocyte and oocyte and so on, right? Okay, so after puberty, this primary follicle gonna get converted into secondary follicle, but still inside this secondary follicle you are gonna get this arrested form of primary oocyte. Now gradually under influence of luteinizing hormone and follicle stimulating hormone called gonadotropic hormones or gonadotropins coming from anterior pituitary, they will resume their uh, cell division and follicular maturation, right? And the secondary follicle will get converted into tertiary follicle, and inside the tertiary follicle for the first time you can observe the next step of primary oocyte, that is called as secondary oocyte upon completion of that particular division. Okay, upon maturation of tertiary follicle, you will be getting the matured form of follicle, that is called as Graafian follicle. Graafian follicle is the matured form of ovarian follicle. Inside the Graafian follicle also you are going to get this secondary oocyte stage, and at that particular time this Graafian follicle will be ruptured and it will throw the secondary oocyte outside the ovary, and that phase is called as ovulation. Upon ovulation, now this secondary oocyte will wait for 12 to 24 hours for sperm to come and fertilize. Now this ruptured Graafian follicle will get converted into a yellow colored body, called as corpus luteum due to the accumulation of lutein pigment, and this corpus luteum will act as individual endocrine gland inside this ovary. The corpus luteum can secrete progesterone, the corpus luteum can secrete estrogen, the corpus luteum can secrete relaxin and many other hormones. And gradually when the corpus luteum will also get matured, and gradually it will die and vanish by forming a white colored body called as corpus albicans. So these are different stages through which we can observe the follicular development inside the ovary. Okay. Now our motto is to study the process of oogenesis in detail. Okay. Now assume this is the embryonic life of a girl child, and inside her ovary you're gonna get millions of oogonia. Now what about this oogonia? They are the precursor cells, and they are also diploid in nature with 46 chromosomes. Now during the time of embryonic life itself, this oogonia will start mitosis. They will form more oogonial cells, and few of the oogonial cells will differentiate. So it's written as differentiation. Differentiation means the cells are gaining mass, the cells are changing their shape. So upon differentiation, few of the oogonial cells will get converted into primary oocyte, right? Primary oocytes are also diploid cells because they are resultant cells of mitosis and differentiation with 46 chromosomes. Now during the time of embryonic life itself, these primary oocytes will start a new kind of division, that is called first meiotic division. So it will start M1 means first meiotic division, but you know that this is not right time for the process of ovum formation. So few of the body chemicals or agents will come to this primary oocyte from the surrounding follicular cells. Now this primary oocyte, you know, it's encaged inside that primary follicle. So this follicular cells will send some chemicals, some signal, and it will forbid the primary oocyte to complete the meiosis 1, and that will be getting arrested, means the meiosis 1 is paused now at prophase 1 phase. So where these primary oocytes are arrested inside the ovaries, right, at the meiosis 1 stage. And which phase of meiosis 1? At prophase 1. And particularly if you peep inside, you will be finding prophase 1 has multiple stages. It is arrested into the diploteny phase. Okay. Now, as such condition, it will remain up to the birth and even up to the puberty. Now, upon arrival of puberty, that is post-embryonic life, right? Upon arrival of puberty, this paused or arrested primary oocyte will get signal from some pituitary hormones called as luteinizing hormone and follicle stimulating hormone. Together we call them gonadotropins. Now under influence, again the meiosis 1 will be resumed, will be continuing to produce a large cell and a smaller one cells. The larger cell is called as secondary oocyte and the smaller one is called as first polar body. The secondary oocyte is haploid in nature with 23 chromosomes. Similarly, the first polar body is also haploid in nature with 23 chromosomes. But why they are varying in shape and size? It's because while the body was performing or undergoing this completion of first meiotic division, that is M1, that will be performing equal karyokinesis, equal nuclear division, but unequal cytokinesis, means unequal distribution of cytoplasmic component. So most of the cytoplasm will be coming inside the secondary oocyte, making it a larger cell, and very negligible amount of cytoplasm will be getting inside this first polar body, making it to a smaller cell, and gradually it will die. Right. Now you may ask a question here, sir, why this kind of uneven or unequal cytokinesis is happening here, which is unlike other cells? Okay, the reason is this secondary oocyte in coming days, it will become a mature ovum upon fertilization, and it needs to provide the major sources of energy. That's why it has to retain maximum of the cytoplasm and the energy resources. And you know one thing, that when it got arrested during the time of embryonic life, it has to wait for 12 to 13 years to resume the process of meiosis 1. In that time duration, it will be losing its one of the important cell organelle, that is centrioles. And you know that for animal cells, centrioles are responsible for equal cell division. If there is no centriole inside the cell, so there will be unequal division of cytoplasmic components. So absence of centriole is responsible for this unequal cytokinesis and formation of a large secondary oocyte. Now you need to focus on secondary oocyte because gradually the first polar body will die. Now at this secondary oocyte stage only, your ovary one of the ovary will be ovulating this, will be kicking this out from ovary, right? And if there is no fertilization, this secondary oocyte will die inside the body, right? But if sperm is arriving, for fertilization, so what happens? This secondary oocyte will start second meiotic division or meiosis 2 here. But again, prior to the fertilization or prior to the arrival of sperm, it got arrested or paused at the phase of uh, second metaphase, right? Now, if sperm is arising, if fertilization is taking place, if sperm is arriving, fertilization is taking place, then only meiosis 2 will be completed. Then only meiosis 2 will be completed, and the secondary oocyte will complete the division to form a larger oocyte or ovum and a smaller second polar body, which will be degenerating in near future. And there will be fusion between the sperm and this oocyte or ovum, and that will form a diploid zygote, right? So as because this secondary oocyte is haploid in nature and the meiosis 2 is equational division, the oocyte or ovum that has also only 23 chromosomes into it. So what I wanted to convey here, secondary oocyte, focus on this particular stage of ovum. Secondary oocyte is ovulated. Now upon ovulation, it will start first, it will start second meiotic division or meiosis 2, but it will get arrested again. Arrested means the division is stopped at which particular phase this will be arrested at a particular phase of called second metaphase. And after that, what will happen? If there is no fertilization, the secondary oocyte will die in such arrested condition. And if fertilization is taking place, while the sperm is taking entry penetrating inside the ovum, there will be completion of meiosis 2 and the secondary oocyte will be divided to form a mature ovum called as oocyte and a second polar body. And from this oocyte, gradually upon completion of the fusion, zygote will be produced. Okay, so this is the structure of secondary oocyte. This is inside the cell. This is the haploid nucleus of secondary oocyte, and it is covered by three membrane. The plasma membrane of secondary oocyte is called as vitelline membrane. After vitelline membrane, outside the vitelline membrane, there is a very important covering of secondary oocyte called as zona pellucida. The zona pellucida is responsible for sperm specificity. With special receptor, it will be recognizing the human sperm, and the outermost layer of secondary oocyte is called as corona radiata. They are nothing but some follicular cells attached outside the zona pellucida layer. Okay, and the space between the cell membrane or vitelline membrane and the zona pellucida is called as perivitelline space. Okay, now we will try to differentiate between the process of spermatogenesis and oogenesis. So first thing, you know that the spermatogenesis initiates during the time of puberty, but oogenesis initiates during the time of embryonic life. Second thing, spermatogenesis is a continuous process. But for the whole process of oogenesis, it's a discontinuous process. There will be two arrests into two different zones. One, from primary spermatocyte to secondary spermatocyte, there will be one arrest into the first meiotic division. During the time of first meiotic division, the phase 1 arrest, and second arrest from secondary oocyte to ootid. There will be another arrest during the time of metaphase 2 or second metaphase. So that's a discontinuous division. For spermatogenesis, we can observe two differentiation phases. One differentiation from spermatogonia to primary spermatocyte, another differentiation from spermatid to spermatozoa. But in case of oogenesis, there will be only one differentiation will be taking place. One differentiation means only during the time of conversion of oogonia to primary oocyte. There will be no further differentiation because here ootid gonna be the matured ovum. So that was the difference between this process of spermatogenesis and oogenesis. So with this we can conclude the lecture. And one more thing, when you talk about sperm or spermatozoa, you'll be finding the sperms are swimming inside a special nutritious fluid called as seminal plasma, right? Together, this sperm and seminal plasma called as semen, and during the time of one ejaculation, 2 to 4 ml of semen is ejaculated by male during the time of insemination. And this 2 to 4 ml of semen contains 200 to 300 millions of sperms. And if that particular male is seeking for proper fertility, 60% of those sperm should be of normal shape and size, and 40% of those should have vigorous, means very fast motility. Very fast moving, 40% of those sperm should be a very fast moving sperm. So that's all. And you know that sperm, they're produced inside the testis, and the seminal plasma, they're produced by the secretion of a special glands called seminal vesicles, prostate and bulbourethral glands. Now let's check few of the questions. And the first question is given: your first haploid cell of spermatogenesis is. The first haploid cell gonna be secondary spermatocyte. Next question, one assertion and reason: the ovum retains most of the cytoplasmic content of the primary oocyte and is much larger than spermatozoa. Obviously, this is true if you have studied oogenesis in a proper manner. And the reason said that the ovum needs energy to actively seek out the spermatozoa. No, that's gonna be a false statement. The sperm seeks energy to reach up to the ovum and penetrate inside it. So its assertion is true, but reason is false. Next question. Seminiferous tubules contain dash cells for providing nourishment or nutrient to the sperm. We call them Sertoli cell, or you can also call them nurse cell. So with this, I'm ending this particular session of gametogenesis. Bye bye and Jai Hind.